first the last point of the last chapter, and then we'll answer together the 2 question pages I've sent, and then go on with the next chapter. So we've discussed last time different procedures for purifying or sorting cells and purifying their organelles. And we mentioned in this context this method, the FACS, for sorting cells. And we mentioned rough centrifugation for isolating cellular organelles, and then a more refined version of the same thing using a sucrose gradient. And the last thing I wanted to mention in this context is this rather elegant technique which allows us to isolate vesicles. So vesicles are relatively small organelles, and instead of centrifuge them to death, one can use the following concept. We can use a bacterial cell, which is a pretty large component compared to this vesicle, linked with an antibody, that will recognize some antigen or will recognize these vesicles. And now these bacterial cells, which are, as I said, are pretty large one, they will sediment quite rapidly when centrifuging, and taking together with them these vesicles. So this is a method first to centrifuge the vesicle in its full shape, an intact vesicle, instead of destroying it while centrifuging it. And the second, you can also try to target for a specific subpopulation of vesicles by using an antibody that will recognize these specific vesicles out of the global pool of vesicles. And this is one of these examples in biology where it's really looked like what we thought it would like. So this is the plasma membrane of the bacterial cell, and on it you can see the vesicles linked upon it via the antibody.
The bacterial cell does, you said that there is an antibody.
Right, linked to the bacterial cell.
Mm-hmm.
Yes.
And the antibody ties the vesicula?
Yep. And the antibody recognizes the vesicle, and it's not a covalent bond, but it's a stable enough bond to centrifuge for the bacterial cell to be sedimented together with the vesicle. So effectively, you just increase the mass of the vesicle. This is, the principle that underlines this procedure. Okay, any questions about this method? Well, I'm not sure. I believe the question is whether natural occurring bacteria cells would have antibodies that can recognize specific vesicles. I guess most of the time they should be genetically engineered to synthesize and to express the desired antibody right here. Okay, and that's that for the architecture and biomembrane of cells. And now let's go over the 2 question pages, starting with the previous one, actually. Molecular genetic mechanisms. We had 2 questions right here, so let's start with them. Okay, so below are several kinds of molecules. Matched to each one, the role, or more than one or roles, it performs in the cell using the following list. And you're given here a list of roles. So let's start with the first role. Decoding of a nucleotide sequence is performed by which molecule right here? The transfer RNA, right? The tRNA. Part of the ribosome is what? Ribosomal RNA, rRNA. Regulation on gene expression is performed by 2 molecules that we haven't mentioned, actually, because we took them out from this chapter, and we're going to learn on them later on. These are microRNA right here, microRNA and siRNA, short interfering RNA, and we will get to know them in the future. 4, part of the spliceosome is, again, I'm not sure we've mentioned them yet. These are snRNA, small nuclear RNA. And the last we have mentioned, so Information storage is the main task of which RNA molecule? The messenger RNA. Information storage or transfer. Decoding is when you take the nucleic acid sequence and decode it, and code it. No, it's like it's the transformation from nucleic acids to amino acids, which is the translation step, not the transcription step. Okay. The second question, in the DNA molecule, so which of these sentences are correct? The strand's orientation is anti-parallel, so that's correct. So B is not correct. And C, covalent bonds hold the two strands together. So which bonds? Which are also called Watson-Crick bonds, right? Specifically, this type of hydrogen bonds are called Watson-Crick bonds. So A and C, which is E. A and D, sorry, which is F, obviously, sorry. Okay. And now for the biomembranes and cell architecture question page. So the first one, some organelles in the cell are surrounded by two membranes, and in between these membranes there is an intermediate space. And the question is, who are these organelles? So what do you think? Mitochondria and the nucleus, which is B, correct. So here it's B. Second question, phospholipids are distributed in a heterogeneous manner between the two layers of the plasma membrane. How does this heterogeneity achieved? So the question if these two explanations can be the reason for the heterogeneity in phospholipid distribution. The first one is different organelles in the cell synthesize phospholipids which harbor, sorry, there should be an H here, which harbor in one of the two layers. So what do you think? Yeah, that's a reason we've mentioned. And the second one is different enzymes between the two sides of the membranes cut different bonds in the phospholipid hydrophilic region. What about this explanation? It's also correct. Which enzymes am I talking about right here? Phospholipases, right? The ones that cleave lipids in certain region across them. So these two explanations can be the reason for the heterogeneity in phospholipid distribution, and the correct answer is A and B are correct. D. 3. Below is a list of cellular organelles. Match one organelle or more from the list to its role in the cell. So this is just, following what was 8 or 10 slides we had in the last presentation. Well, let's start with here. Degradation of outside material and all cellular organelles is performed where? In lysosomes, correct. ATP synthesis by glucose oxidation. Mitochondria is the one that performs ATP synthesis and production. Synthesis, processing, and sorting of secreted proteins. The RAF-ER, RAF endoplasmic reticulum. Storage of secreted proteins is secretory vesicles, right? And last one, regulation of entry and exit of molecules to and from the nucleus is the nuclear membrane is the one that performs this task. Okay. Okay, so for our next chapter, transport of ions and small molecules across cell membranes, which is largely parallel to the 7th chapter in our textbook. So we've discussed the structure and characteristics of biomembranes, and we will now see the different transport mechanisms that exist to transport molecules across these barriers that we've learned about. It's a relatively large chapter, so we will start with an overview of membrane transport, then proceed with or focusing on ATP-powered pumps and see how they determine the intracellular ionic environment. After that, we will learn about non-gated ion channels and see how they contribute to the resting membrane potential. Some molecules or some transport proteins can transport more than a single molecule. They can transport in parallel 2 or even 3 molecules. These are called transporters, co-transporters, sorry. We will say just a word about the water pump or the water channel aquaporin. Then learn about the voltage-gated ion channels in opposed to the non-gated ion channels. Okay, so gated ion channels can be gated by several different stimuli. They can be open and closed by a mechanical stimulus, like mechanical receptors in our skin. They can be open or closed by an electrical potential, recognition of specific ligand, like neurotransmitter. And we will focus in this class on the voltage-gated ion channels. And we will see that they are the one that enables propagation of action potentials along the long projections of neurons. And we will end with learning about some major components in the synapse, like neurotransmitters, synaptic receptors, and synaptic transporters. Okay, so most molecules that exist inside the cell cannot pass by their own the hydrophilic barrier that is the membrane. Some exceptions for that rule is gases, for instance. So CO2, N2, and O2 do have almost a full permeability across the cellular membrane. Also, small, uncharged, but polar molecules such as ethanol, urea, or water have partial permeability to the cellular membrane. So water, for instance, does have a specific channel right here on the membrane, but it can also pass partially or under some conditions just as it is through the membrane. But for most of the molecules, this is not true, and they have to have a specific active mechanism, transporters, channels, what have you, to allow the transport across the membrane. And this is true for large uncharged polar molecules, charged polar molecules, all different ions that exist in the cell. Because of their polarity, they have to be able to cross via some hydrophilic tunnel inside the hydrophobic barrier that is the membrane. And so they have really no, they are totally impermeable across the cellular membrane. Okay, all transport proteins are transmembrane proteins because they have to form a hydrophilic buffer zone, we can call it, inside the hydrophobic plane of the membrane. So they have to be transmembrane, and most of them would have alpha helices or membrane-spanning alpha helices as the stabilizing structures in the protein bodies. And we've already mentioned an important specific structure in this context, the 7 membrane-spanning alpha helices structure that is found in many, many transport proteins. Okay, and the way they transport molecules is by a series of conformational changes. And we will see specific examples for this principle. Okay, so these are the 3 classes for transport proteins that exist. We will go in depth in each of these in a short while, but for now, just appreciate the transition rates of each of these classes. So ion channels are the fastest. They move around 10 to the power of 8 ions per second. And that is because a single conformational change in these proteins equals the transition of many ions. In opposed to transporters and ATP-powered pumps, which in these cases, each conformational change equals the transition of just a single molecule or just a couple of them, just several molecules. So their transition rate is about 10 to the power of around 1,000 molecules per second. It can be a bit more than that, but that's the average. Okay. So let's go into the ATP-powered pumps first. okay, these transporters use the energy stored in the ATP molecule by hydrolyzing it. And they couple this energy to the transition of a molecule against its concentration gradient. So each time this triangle will denote, will represent in this class concentration gradient, meaning that this red ion have high concentration in this side and low concentration in this side. And ATP-powered pumps hydrolyze ATP, thereby coupling this exothermic reaction together with this endothermic reaction. Because moving an ion against its concentration gradient is an unfavorable reaction. It's a reaction with a positive delta G or an endothermic reaction. Okay, all of these pumps are, as all other transport proteins are transmembranal, they would always have an ATP binding site in the cytosolic side of the protein, yeah, right? Because they have to bind ATP and hydrolyze it to have the energy to transport the ion. And they are the ones that responsible for the generation and maintenance of ionic gradients across cellular membranes. And this is the typical standard picture of ionic concentrations inside of the cell and outside of the cell for almost any cell that exists in nature. So ionic concentrations or ionic gradients across the cellular membrane is not a feature that is specific to neurons. Okay, neurons are cells, and as such have some mechanisms in common with all other cells. For instance, this feature, and they have some specific features only for neurons. So only neurons produce rapid disruptions in the ionic gradients, which are actually the action potentials. But these gradients are produced and maintained in all living cells. All living cells have to have these gradients. And you don't have to remember all this table by heart, but for the three first ions, potassium, calcium, and chloride, potassium, sodium, and chloride, you do have to remember in which side of the membrane do you have a higher level. So it's quite easy to remember. Only potassium has high levels or high concentrations inside the cell, and sodium and chloride are in high concentrations outside of the cell. So if we if we allow permeability, specific permeability for sodium, for instance, the driving force will be towards the inside of the cell, and vice versa for potassium. And these are exactly the ATP pumps that generate and maintain these gradients, which are so essential for all living cells and for the production of action potential in neurons and so on. And just to, a way to appreciate how important is this phenomenon of ionic gradients is by considering the following fact. So up to 25% of all ATP produced in neurons and also kidney cells, well, kidney cells are not the major focus of this class, but kidney cells filter salts in the body using transport proteins on their membrane. So they are, they have very similar features in this respect to neurons. So about a quarter of the total ATP production is dedicated to transport of ions across cellular membranes. Just try to think how many additional processes are in cells, not only, together with transport of molecules. So that's a huge fraction of the ATP production. There are 4 classes in nature of ATP-powered pumps, and we will only mention the first 2. I've brought the last 2 just to give the full picture. So the first one is the P-class pump, which is really very, very much conserved across the evolution. You will find this protein in the cellular membranes of many, many kingdoms in nature. It has all the proteins that belong to this class has a similar operational mechanism, and we will see here the operational mechanism specifically for the muscle calcium ATPase. But by that we will, learn the principle of all P-class protein operational mechanisms. So so the muscle calcium ATPase pump is to pump calcium into the sarcoplasmic reticulum in muscle cells. The sarcoplasmic reticulum is a specific type of endoplasmic reticulum, something that is thought to evolve from the ER, and that is used in the cell as a huge storage of calcium ions. So inside the SR lumen, the calcium concentration, so lumen, you remember, it's the aqueous interior of the SR. Inside the SR lumen, you'll find high concentrations of calcium, but this pump transport an additional calcium against its concentration gradient using hydrolysis of ATP. So everything starts with the binding of calcium from the cytosolic side to two binding sites in the protein, together with the binding of ATP to its binding site. Next, ATP hydrolysis. The lone phosphate is linked here to an aspartate residue. This is also a conserved feature in P-class ATP pumps. It will be always the phosphorylation will occur upon aspartate residues. And now this reaction we've just gone up the energy level of this protein, and that's exactly the energy that will be used to drive the conformational change between E1, where the two calcium binding sites face towards the cytosol, and the second conformational state, in which the two calcium binding sites face the lumen of the SR. Okay? So this is where the conformational change occurs right after the ATP synthesis. In this state, the affinity for the calcium of these two binding sites decreases very substantially, and the two calcium ions are released into the SR lumen. Dissociation of the lone phosphate from its binding site brings us back to the first step, where the protein is again ready to bind to new calcium ions.
Which is the membrane of the neuronal reticulum?
Of the sarcoplasmic reticulum. Yep. It stands on the membrane. What's that?
Oh, inside the cell.
It's inside the cell. Like we know already where the ER is located, right? It's definitely inside the cell. And it's a specific organelle which we haven't mentioned, but very much similar to the ER in muscle cells. So, and now the reason, okay, so it's not essential for our class, but again, calcium, when it exists in high concentrations in the cytosol, it performs many tasks. It binds to proteins and it activates many processes. So you have to buffer it very quickly inside into some storage place, and that's exactly what the SR lumen is for the calcium. Which error? The triangle. So the base of the triangle is where you have high concentration of a specific ion, in our case calcium, and the tip is where we have low concentration. It denotes the concentration gradients towards the cytosol. Okay, and this is how the muscle calcium ATPase looks like in a water-accessible surface model, and in the more simplified cylinder and trace backbone model. So this one is the ATP binding domain, and the phosphorylation domain, again, specifically, it will always occur upon an aspartate residue. And the transmembranal domain is where the two calcium binds. This is the E1. This is the E1 state, where the calcium binding site faces towards the cytosol, and upon hydrolysis and the conformational change in this protein, there's a major dislocation of the ATP binding right here, the orange domain, which drives with it a conformational change up here in the transmembranal domain, which then causes the binding site of calcium to face towards the lumen of the SR. Okay? So that's very briefly how things look like on the structural level. Okay, let's see another example, this time for what is called the sodium-potassium ATPase powered pump. And again, the name is always that the molecules that participate in or they are being transported. And ATPase means that it's an ATP powered pump. Okay, so this time everything starts. Let's start with the end. This pump generates and maintains the following concentration gradients. Sodium is high outside the cell and low in the cytosol, and vice versa for potassium. We've seen this for the typical picture of an ionic environment in the table at the beginning. So this time we start, it's a very similar mechanism. In the E1 state, where you have high affinity for binding site, for binding of three sodium ions in these binding sites together with ATP, hydrolysis, phosphorylation on an aspartate residue, which gives us the energy required for the transition between E1 and E2, right here, the conformational change. In E2, the affinity for the calcium ions, for the sodium ions, sorry, right here, the affinity decreases very sharply. The 3 ions leaves the binding site. 2 potassium ions then have high affinity to their binding site, and they're indeed linked there. And okay, we're still in E2. And now dissociation of the lone phosphate right here from the aspartate residue is what drives us back to E1, where the affinity for the potassium ions again decreases very sharply, and the 2 potassium ions leaves to the inside of the cell. And the net reaction is exactly is what gives us this picture, and is more specifically 3 sodium ions are taken outside and 2 potassium ions are taken inside. So what would be the net charge? Would there be any accumulated charge in one of the sides of the membrane if you will only consider this pump? So you take out 3 positively charged ions and take in just 2 positively charged ions, right? So there's an extra of it, let's say it like this, there's a delta negative charge that is being left in the inside. So this is an example of what we call electrogenic channel, or electrogenic pump in this case, electrogenic transport protein, which have a net charge in its reaction that is different from 0. So just remember this term, and we will be using it soon. Skip the movie, okay. For the V-class proton pumps, we will not see the specific operational mechanism, just mention its main role in the cell. So these pumps are found, are very common to be found on endosomes and lysosomes, and they are using the ATP, the energy stored in ATP, to transport protons into endosomes and lysosomes, and thereby acidify their lumen. Right? More protons in the lumen of the endosome means more acidic pH. Okay, and the thing is this: if this pump would start to transport protons into the lumen of the endosome, then only after transport of a couple of protons, and you would already there will be a buildup of a very large electrical gradient right here, because each proton carries with it a positive charge, which will effectively leave us with a delta negative charge in the outside, and here electric potential will be formed very rapidly. Very rapidly, I mean by transport of only several proton molecules. So this is also an electrogenic example of transport protein, and it has to be coupled in order to allow sufficient acidification of the endosome lumen. It has to be coupled together either with movement of a positively charged ion towards the outside of the endosome, or with a negatively charged molecule to the inside of the endosome, which is what we see here. So it's enough to open a chloride channel to allow, or to destroy, to neutralize this electric potential, and to allow sufficient buildup of protons inside the endosomes.
What would be if it would be some electric potential?
Again, sorry?
What would be if there will be an electric potential?
Well, this electric potential is a force that is opposing any additional transport of protons inside the endosome. It's the electric. So each ion feels really 2 forces, right? It feels the chemical gradient. It feels a need to flow down its concentration gradient chemically wise, and it feels an electrical potential, which means just electrostatic repulsion or electrostatic interaction with with positive or negative charges. And it's the electrical force that builds up much faster than the chemical force. And we will see it again later on. So again, it's sufficient to change the chemical gradient of the protons just a little bit in order to build up such a huge electrical potential that would oppose any additional transport of protons. And so you have to couple it together with the movement of a chloride in this case, which is most commonly what happens. Questions?
In the PACCR with the potassium and sodium, there is also a potential.
Right. And in this, definitely, this is also an electrogenic channel. Not channel, an electrogenic pump. And so again, this one, I just isolated this process, but coupled to it and outside the scope of this slide, you definitely have a movement of additional ions in and out of the cell. Yes? Yeah, what part?
After the electric potential is created,
Yes.
what will need to happen and for what?
So the electrical potential in this case is just, you can address it as an artifact just because you pass proteins inside the endosome lumen, protons inside the endosome lumen, and so this gradient is formed. So this part is understood. And then in order to this gradient is opposing any additional transport of additional protons inside the endosome, and so you have to neutralize it some way, and the most common way would be to allow the entrance of chloride into the endosome, which will free or allow additional protons to enter the endosome, and for its lumen to acidify sufficiently. Okay, so that was ADP-powered pumps and the way they control the intracellular ionic environment. Let's go on with non-gated ion channels, and we'll see how they contribute to the resting membrane potential. So first, channel proteins transport ions down their concentration gradients. This time it's down the concentration gradient. And now, not to insult anyone, but these are really just holes in the membrane. Maybe selective holes, but they are just holes. So they can only allow the transport of ions to where they want to go anyway. So it will always be down the concentration gradient. They have essential outcomes in living cells, but still they only pass ions down their concentration gradients. And they can come in two flavors. There are some channel proteins that have a gate, which is regulated by some feature, and there are some which are they are always open. Effectively, they have no gate. So as long as they are located on the membrane, they are open. So in these examples, the non-gated ion channels, what do you think? How would the cell regulate their function? For instance, if the cell needs less potassium at a given moment, and there are plenty of non-gated potassium channels in the cellular membrane, how would it regulate the transport of this ion? Sorry? Denaturalize? Neutralize the channel, but how? I mean, it doesn't have a gate. So how would it do it? Okay, so just remove the protein and then degrade it somewhere in the cytosol. But just the removal of the protein will be the only regulation process in which you would regulate, so to speak, these channels. But again, as long as they are in the membrane, they pass ions. That's important to remember. Okay, and different non-gated ion channels exist with the selectivity for specific ions, and they can pass ions in different rates. And these are the pumps that we've just mentioned, the ADP-powered pumps, together with the ion channels, the non-gated ion channels that are responsible together, again, for the electric potential across the plasma membrane, which is not only for neurons but for all living cells, is about minus 70 millivolt. Okay, so how would this, how exactly, what's that? Should be a minus right here? Yeah, if it's not, it's just the potential with no direction, so I don't think you don't have to write a minus. Never mind, let's add a minus there. How exactly are this electrical potential is generated? Simply by introducing selective holes in the membrane. So the explanation is the following one. And for it, let's consider a tank filled with water, having two cells or two compartments, with an impermeable membrane or a barrier in the middle. So the first cell is really equivalent to the cell cytosol, and it has low concentration of sodium compared to the second compartment, and high concentration of potassium compared to the second compartment. And that's where we start our explanation. In this case, we read no membrane electric potential. The potentiometer would show 0. Now it's enough to add specific selective non-gated ion channels, for instance, for sodium, here in the membrane, which will cause an influx or an inward current of sodium into the left cell, the left compartment, until what? So if you open these holes here, sodium would start to flow until what would happen? Equilibrium with what? With the electrical gradient. Okay, so each ion here feels two forces, or two driving forces. The one is to go down its concentration gradient, but at a certain point, the extra or the excess of positive charge right here would oppose any further current of sodium into the left cell. Okay, and in this case, you see that the membrane is depolarized. There's an excess of positive charge, and on each sodium that enters the cytosol, we have an effective delta charge that we leave outside, and we get, and then an electric potential is formed across the membrane. And the potentiometer would show some value different from 0. So again, it's enough to introduce, when you have this situation of different concentration gradients across the cell, it's enough to add non-gated ion channels to explain the formation of electrical gradients. Yes?
So if you're adding a lot of positive sodium ions, why does it become negative?
The outside becomes negative because the positively charged sodium ions enters the cell, and so relative to what was the state beforehand, so there's a delta charge that is being left behind, effectively speaking. Okay, now what would happen if I add here several chloride selective non-gated ion channels? I would add here holes that will allow the passage of chloride. We know what, just ignore for now the concentration of chloride, but still just, so what would happen? Yes, let's, individual. Which one will go the other way? The chloride. But the chloride will go against its, well, if we'll only consider, let's ignore the concentration gradient right now, but if we'll consider only the electrical potential, then the chloride will want to go towards the oppositely charged region, which is towards the inside, and we might reach a point where the electrical potential will be abolished or neutralized, right? So, and this is exactly what stands in the base of, or this is exactly the molecular explanation for the minus 70, because different non-gated ion channels exist in the cellular membrane, but they have different rates of transition, and also they exist in the cellular membranes in different abundances. So the most abundant non-gated ion channel is for potassium, and the battery or the reverse potential for the potassium ion, and I've just used the term that we haven't discussed in this class, the battery of an ion, so just read about it at home, please, is around minus 80, and that's the main determinant for the electric potential of cellular membranes in a basal state, when you don't have no action potentials or anything. So the most common non-gated ion channels that exist in the cellular membrane is for potassium. That's, again, it's a fact that is very important to remember. And because potassium are in high concentrations inside the cell, they would want to leave the cell, leaving a delta negative charge inside the cell. Hence, we have an inside negative situation in living cells. And specifically, we have minus 70. The minus 70 comes exactly from the Goldman equation, which takes into account the different permeabilities for the different ions and their batteries. Or to be more precise, the difference between the batteries and the current voltage for the current potential of the membrane. So read also just a couple of sentences about the Goldman equation. Sababa. Yeah, ion batteries. These ion batteries, also called the reversal potential. Okay, let's see in more details the structure and the function of the potassium non-gated ion channel because of its very essential role for determining the minus 70 millivolt membrane potential in living cells. So it's a tetramer built of 4 identical units, so it's a homotetramer. Each subunit has 2 alpha helices linked together by a loop, which more specifically have some region in it that is, or the pore, the P here stands for the pore, because it's the pore segment, because it bends into the cavity of the channel. You can see it right here. This chupchik here is the P segment, and inside it, this small fraction of polypeptide is exactly the selectivity filter, and we will look at it in zoom in the next slide, exactly on this region. So just remember where the selectivity filter stands in relation to the single subunit and in relation to the total structure of the protein.
What did you say the P stands for?
Pore.
Pore?
Pore. Teala, channel, tunnel, pore. Okay, remember another term, the vestibule is... Sorry? How, again? The P segment, the P segment bends from, okay, so again, these 2 alpha helices are the transmembranal regions of the protein, the 2 yellow alpha helices, and between them, the P segment linked them both and bend into the cavity of the protein. So if I am an alpha helix, this would be the P segment that's bended into the cavity or into the pore. So that's the reason why we call it the P segment. Okay? And again, a small fraction from it serves as the selectivity filter for potassium. Okay, and that will be so we will continue with the next slide, which is directly related with this one, and after that we'll take the break. We are now zooming into this region right here. Yeah. Okay. Okay, it was a long-lasting question in neuroscience. How come the potassium ion channel allows passage of potassium ions and not allow passage of a smaller ion, for instance, sodium? It's very intuitive to think why the potassium ion that is designed for the size of the potassium ion would not allow any transport of ions that are bigger than potassium, right? Because effectively, channels will not pass any spheres that have a diameter bigger than that of the channel, okay? But if there are smaller spheres, so how come they are not passing at all through the channel? And now for the explanation. So when the potassium ion passes through the non-gated ion channel that is selective for potassium, it interacts with the 4 oxygens that are the carbonyl oxygens from the residues glycine that stands right here in the selectivity filter. Here you have alternating glycine and tyrosine, and the oxygens, the backbone oxygens of these residues, interact with the potassium ion while it passes the channel. And they interact with it in a structure that mimics the interactions of the oxygens, of the water oxygens, when the ion is outside the channel in a hydrated form. So as you recall, when ions are in aqueous solution, they are surrounded by a shell of water molecules. Specifically, potassium ion would be surrounded by 8 water molecules. We are only looking at 4 right now, and there are an additional 4 underneath these 4. When the potassium ion moves from the solution to the inside of the channel, it switches its interactions with the oxygens from the water molecules with the interactions which are really exactly the same, meaning the same distances and the same characteristics. It switches its interactions with the oxygens from the residues in the selectivity filter. So this transition is smooth, so to speak. It has a low activation energy, and that's why it happens. For the sodium, on the other hand, the distances or the nature of the interactions between the sodium and the carbonyl oxygens in the selectivity filter are not the same, and quite different actually from its hydrated form when it's outside the channel. So this transition has high activation energy, and hence just doesn't effectively happen in very minute amounts. So it almost doesn't happen simply because of the high activation energy, which is the result of the different interactions between this state and that state. And to end this explanation totally, so let's do another slide. So this is a kind of a front view. We are sitting at the channel and looking up the ion while it comes towards the channel. Now let's take a side view, and it's a side view. We only see 2 units here. It's a tetramer, right? So the additional 2 units are inside the board and outside the board, and I've just sliced the protein, and we're looking at 2 units. So while the potassium ion passes the channel, it interacts with 8 oxygen carbonyls, with 8 carbonyl oxygens, sorry. Here we only see 4 because we see just 2 units. It interacts with 2 carbonyl oxygens from each unit. So in total, we have 8 interactions, which is exactly the state in the hydrated form. So here, for instance, you see a potassium ion that interacts with 4 water molecule oxygens and 4, 1, 2, inside the board and outside the board, carbonyl oxygens, which is the transition state. Okay, now this channel passes plenty of ions in any given second, as we saw. And more specifically, when plenty of ions pass through this channel, they do so with 2 alternating states. State 1, because each time you have to have 8 interactions for each potassium ion for it to be stabilized. And so, although there are actually 4 positions where the potassium ion can stand inside the selectivity filter, in any given moment, you would only have 2 potassium ions, right here in position 1 and 3, and then in 2 and 4, because this is the only way where each potassium ion gets is 8 interactions, and no one fights on the carbonyl oxygen interactions. Yeah, just let me make sure. Okay.
The selectivity is simply because of the distance between the P segments?
Yeah, exactly.
And the coding is always at the same
distances. Yeah, exactly. So the potassium, there's something very stable, structurally stable that holds, somewhere outside this picture, but that holds these 2 segments in specific interactions, in specific distances between these 2 carbonyl oxygens, which is designed for the potassium ion and not for the sodium ion. Okay, any questions? So that was the structure and function for non-gated potassium channel. Okay, let's take a break, 15 minutes, and then proceed. לא, כי כאילו זה, עכשיו שניהם רבים עליו, כן. אז זה כאילו, כן.
טוב שהוא חוזר קצת על החומר, אני חושב שכדאי לספר.
אני חושבת שהם בנו את זה בכוונה ככה שזה יחפוך.
גם בזה של פורום של דנה, אנחנו רגילים על זה, לדעתי צריך להשתמש באחד מהם, באחד התרגילים צריך להשתמש, זוכר שאמרנו שפאי זה כפל זה כמו סיגמה לחיבור?
פאי לכפל?
יש לנו סיגמה לחיבור,
כן.
צריך לעשות פאי לכפל?
כן.
יש שם מקום שצריך להשתמש בזה.
בייחוד החדר, כאילו, כן. שנים, את נשואה כבר 30 שנה.
כבר יותר משנה, זה לא...
שנה מחדש, גוזלים בעולם.
אני בגלל שאני מזרח ובדברים אחרים. עברנו את השלב הזה.
כן, כבר לא שם. לא הרבה מאז. חזרת שבוע. מה,
לא הספקתי לקרוא.
כן, אנחנו נפלת את יואב. עשיתי את יואב. השאלה הראשונה אני כזה על חצי, כאילו זה בעיקר פשוט שיגע אותי על לשבת על המשחק הזה מסודר. וזהו, ואני רוצה להשוות אותו תקופה מראש, אני לא הולך לזה, ואני מקווה שהפעם יהיה בסדר או מוכן. פעם זה היה לי
כן, בסדר, אבל גם בא...
לא, ברור.
ונמצא...
לא, סתם, שטויות, לא זה, אבל... זה היה מעצבן התרגילים ומלחיץ כזה. כאילו, והגשה פה, בין הרבה שאלות בסופו של דבר יכול תרגיל, אני אומרת, מה, מצטעני, מה עשיתו למשחק? 4 שאלות, כל מתווה, כאילו טורטנו 4 שאלות. כאילו טורטנו, אני חושבים זה לצורך העניין, כמה שיעורים מכובדים, שיעורים זה לעומק קצת. כאילו, לא לעומק, אבל יותר נחזירים את זה, זה טורטנו, אין התרגילים.
לא דיברנו איתו בסוף על הזה של בריבוע.
נכון, תגידי, מה עם המאמרים של קבלת החלטות? אני לך מה הקטע? אני צריך לחבירו.
אני גם את זה הייתי צריכה אותו בשופט.
זהו, אז... לא, פשוט לא חשבתי לקרוא את מה ששלחתי, זה היה ממש משום מה מהיר. אוקיי? לא, זה לא נורא, זהו, רק...
עידן אמר שזה כן סודי.
לא, זה היה משהו קרי נראה לי, אבל סתם אני מתורטת את ההצלמות שלי. בעיקר לא צריך לקחת כזה הרבה זמן, זה רק כאילו...
זה מבינה, אני בו שנתתי את הדף הזה, אני חושבת שאיבדתי אותו. אני נכנסתי...
So we've seen how ATP-powered pumps generate ionic gradients, and the non-gated ion channels together with ATP pumps are responsible for the resting membrane potential. Let's move on to co-transporters. So, some transport proteins transport a single molecule down its concentration gradient, for instance, ion channels that we've just discussed, which are actually a specific case of a uniporter. Some co-transporters can transport 2 or more molecules, and in this time, if the ATP-powered pumps have used the energy stored in ATP, these proteins use the energy stored in the electrochemical gradient of one ion to pass a second ion against its concentration gradient. So again, one ion will always be transport against its concentration gradient, and another down its concentration gradient. This endothermic reaction is coupled to this exothermic reaction. If the two ions are transported to the same side of the membrane, then the co-transporter is termed symporter, sim for the same side. And if the two ions are transported to both opposite sides of the membrane, so the co-transporter is called antiporter. But in both these cases, one ion is moved against its concentration gradient, and another down its concentration gradient. So that's a different kind of energy coupling from ATP hydrolysis pumps. Yes, they are the best.
What is the energy gradient?
What's the energy? What, in numbers, or what's the principle for? What does it mean? Why would the ion want to move down its concentration gradient? What do you mean by what? Try to be more specific, and I'll try to.
Which energy is released by this downward energy?
That's the electrical gradients or chemical gradients are, as they are energies. They are form of energies, form of potential energies that can be used, for instance, for this transition. I mean, you don't have to have any breaking or reforming of chemical bonds. The gradients themselves are potential energies. Yes. Yes. But the thing is not that it moves from here to there, but that it has a driving force. And that's because of the rule of diffusion down the concentration gradient of an ion. It's like, I don't know, like hot air, is driven towards colder area in the atmosphere. So asking why, it's an empirical law. That was good. Empirical law.
You solved the problem.
You know why they do so? Which energy?
So that's the kind of it's an electrical gradient energy, electrical energy, or chemical energy, or chemical gradient energy. It's a form of energy. It's the same thing as if this object is high, it has some distance from the floor, so it has potential energy. It's enough to push it a little bit, or in this equivalent maybe to open a conductance to a specific ion, conductivity to a specific ion, for this energy to come into play, to be implemented. Okay. More than that, I don't really have better answer. Okay, so these were actually the definitions we just gave for symporters and antiporters. The most common case is where antiporters and symporters use the chemical gradient energy stored in the electrochemical gradient of sodium and proton ions. Okay, so let me ask you already at this point, so first of all, I'm telling you that sodium have a delta sodium have a negative G. Yeah, the transition of sodium into the cell is a favorable thermodynamically process. And why is that? Why is it exothermic? So first of all, what are the concentration of sodiums across the two sides of the membranes? there is higher concentration outside. So due to its electrical gradient,
It comes from the exothermic because energy comes from the outside.
yes. So now no, not exactly that. But I ask why sodium would want, or would strive to go into the cell. So first of all, because of its chemical gradient, and because also the electrical potential, because living cells are inside negative and the sodium is positive, so you have lots of reasons, or two good reasons, for the sodium to go inside. So most commonly, co-transporters would use the gradients of sodium because it's a process with high content of energy. And we actually would not see any operational mechanisms for these proteins. We would just mention the major task, or one task they perform in the cell, and this is pH regulation. So cells have to have specific, concentration of protons inside them, and the ones that control these concentrations are co-transporters. So if we are, this is the pH scale. If we have lots of protons in the cell, meaning acidic pH or low pH, then this protein, the sodium proton antiporter, comes into play. It becomes functional, and it takes out, it uses the energy stored in the downhill movement of sodium to take out a proton, which will push us towards a more basic pH. And in a different region of this scale, another protein becomes functional. This is the sodium bicarbonate chloride antiporter. What this protein does is, it uses the energy stored in the sodium electrochemical gradient to move a bicarbonate inside the cell. That's a basic species. Let's just take it as it is. It caused the inside or its environment to become more basic. And if the cell environment becomes too basic, so this protein becomes functional, and it takes out a bicarbonate, which again, it's a basic species. So taking out the basic species would push us again towards an acidic pH. And the cooperative function of these three molecules can explain the correct regulation of pH inside cells. Yes? Okay. Well, in this case, the chloride is chloride is in high concentrations outside of the cell, and it uses the downhill movement of chloride to take a bicarbonate outside.
Why does it make the inside of the cell more
Acidic. It has, this species interacts with water molecules and other molecules and pushes the environment towards a more acidic nature. That's a basic species. It's a base. This molecule is just simply a base. Okay. Pretty laconic, I know, but that was that for co-transporters. And also not less laconic would be the next point about aquaporine, which only have one slide. But it would be wrong not to mention it at all. So, okay. So we just mention it and go on to more neuronal subjects. So this is the aquaporine, the channel that's specific for water molecules. It's a tetramer, a homotetramer. This time, each unit in the tetramer has a channel, or includes a channel in itself. This is in oppose, for instance, for the non-gated potassium channels we've just seen. We just see its structures, where the combined structure of the four subunits together form a vestibule, or a channel inside or in the interface between the four subunits. And this is different in this case in the aquaporine, where each unit is a channel by its own. Okay, do you see it? Now each unit is not functional as its own in the cellular membrane. They have to be linked together, but still when they are linked and when the tetramer is fully built, then each unit serves as a channel through which water molecules can pass. Now each subunit, if you unfold this structure, each subunit has three pairs of homologous transmembranal regions. These are the pairs are A tag, B tag, C tag. which are connected by two hydrophilic loops. This is the loop right here and right here. And now if you fold the structure again, so these alpha helices are the one that between them the channel is formed, and these two pores, pretty similar to the structure we've seen for the potassium channel. These two pores bend into the cavity and form the selectivity filter, this time for water. Okay? So again, these two components, meta components, I mean the alpha helices, which give rise to the channel itself, and these pore or loops, as they called here, structures which bend into the channel and form a selectivity filter, is a principle that you can see in this channel, and we've seen also for the potassium channel, and actually for many other selectivity, ion selective channels. And when water molecules pass through this channel, they do so via a series of interaction or hydrogen interactions with the carbonyls of the alpha helices of the channel. Again, similar to the potassium channel. Okay, so that was aquaporin. What is it good for? Before my question, your question?
What interaction?
Okay, so for the potassium, we've seen that the potassium have to interact with the carbonyl oxygens. This time it's for the water molecules interact with the carbonyls, which are the carbon atoms themselves. The carbon atoms from the backbone of the polypeptide right here in this region, in the alpha helices region. Okay, so why would the cell, as we said at the beginning, water molecules have partial permeability to pass across the membrane. So why would the cell want to regulate more tightly the amount of water molecules in it? Osmosis is the process, but what the cell gains from this kind of regulation? Sorry? Producing energy by what? By just delivering water molecules here and there. So what the major thing that changes when the cell takes out plenty of water molecules is its volume. Okay, so which also affects the strength of its membrane and so on. But the major thing that changes is the cell volume. So this is the cell regulates its volume by synthesizing and localizing more or less aquaporins and localize them in the cellular membrane. The water molecule, the movement of the water molecule, both sides. So this pore can transport water molecules to both sides.
And how does volume can regulate exit the force to extract the water molecule outside the cell? I mean,
it's vice versa. When you exit, if I understand you correctly, when you take out a water molecule, then or plenty of water molecules, you decrease the volume of the cell.
yes.
Okay, so what was the question?
But how does the water molecule know how to get out? I mean, if the volume, how is the
Down its
volume
constant.
formed?
So just what's so just it goes out down its concentration gradient.
Okay.
Okay. But if
Why did they go?
What is the purpose of the volume change?
Okay, so different. Okay, I can mention a couple of roles, and I think we will also mention them in a couple of slides. So let's wait with this question. But what if the concentration is? I don't think there's a state where the cell. I'm not sure. Okay, what I'm hesitating about is if the cell has high concentration of salts outside, and it likes to take out water molecules, so it goes down its concentration. It's a downhill movement of the water molecules, and that's fine. And if the cell has and vice versa, so again, water molecules will go just down their concentration gradient. It fits.
Can it be that also? Yeah, but can it be also when
Yeah, well, that if enough water molecules would exit or enter, so, but then a whole water molecule carries with it hydroxyl and proton.
Right.
So I think this process is balanced in the pH sense. I think it's quite balanced in the pH sense. I guess it would make some difference. It's like when you dilute something and pH stays the same, because you dilute it with a species that has hydroxyl and protons, meaning base and effective acid together. Okay. Okay, the last point where we will meet the voltage-gated ion channels. Again, we've met non-gated ion channels. Other channels exist that are open upon a mechanical stimulation, upon a heat stimulation, and other kinds of stimuli. And we will focus on those that are open or closed, given a certain voltage, and see how they allow propagation of action potentials along the very far, long projections of neurons till the far terminals of neurons. So an action potential is a series of sudden changes, of sudden interruptions in the ion gradients that we already know how they are formed, and it's really several different channels and pumps are engaged in these sudden changes. So let's first link to what we've already learned. So these are the potassium non-gated channels, and in the basal state, as we said, the inside is negatively charged. And if we will have some accumulation of positive charge in the inside of the cell, due to some reason outside this light, maybe due to ligand binding channels that recognize the ligand, neurotransmitter, open a channel, and for instance, sodium would start to flow in. And in short, again, the inside becomes positive for some reason. If it becomes positive enough, so these, for instance, in this case, the sodium voltage-gated channels would open, would cause additional inflow of sodium ions, which will cause additional positive charge to accumulate in the inside, or additional depolarization of the cell. Depolarization because this is hyperpolarization, and by D I mean destroying the polarization. So it's depolarization, becoming more positive. So they will push us towards a more depolarized state, which will again will cause more voltage-gated sodium channels to open, and that this positive feedback that is caused by the voltage-gated sodium channels is what stands in the first very sudden sharp rise phase in the action potential. So this typical, how the most classic action potential looks like is something like this. And these are the any other color than blue? No. These are the sodium, the voltage-gated sodium channels that are responsible for this rising phase because of the positive feedback we've just learned. Okay. Okay. All sodium and actually all voltage-gated channels have some region in the protein that has high concentration of positively charged amino acids. This is called the sensor domain, the voltage sensor domain. And it's a sensor because as long as the inside is relatively negative, so it stays the same simply because of electrostatic interactions that holds it where it is. You have opposite charges right here, so it feels fine. And if the inside becomes positively enough, then this voltage sensor is just pushed towards the exoplasmic side, and with it carries some conformational change that in this channel cause an opening of a channel, and for this case, sodium currents to flow inside the cell. Okay. After a short while, about 1 or couple of milliseconds, the inactivation domain, which is usually a globular domain in the protein, sticks to the channel opening and blocks any additional sodium current. Now, as long as the inside of the cell is positive, this inactivation globular domain stays where it is and inhibit any further sodium current. now the membrane potential is restored, meaning it becomes again hyperpolarized. So we can call it repolarization process, which is the outcome of the function of other proteins outside this slide. For instance, potassium voltage dependent channels and ATP pumps. And when the cytosol is the negative charge on the cytosol is restored, we're again in a hyperpolarized state. So the globular inactivation domain leaves the opening of the channel, and the channel is again active. This is what we call, or this is the atomic explanation for what is called the refractory period, which is the minimal time period between two spikes or two action potentials. Because after the first action potential, the inactivation domain it sticks right here. And any additional, even if you try to artificially inject positive currents, then this channel is inactivated because even simply because of the positively charged inside of the cell. And it has to be, the membrane has to be hyperpolarized again for this protein to become functional again. One more word. The voltage sensor domain is usually positively charged because of high concentration of lysine and arginine, which are two amino acids with positive charge in physiological conditions. Yes?
How does it explain the refractory curve?
So the refractory period is when one action potential have just occurred, and now you try to depolarize the membrane even further to give rise to another action potential, and for most of for voltage dependent channels, you will not be able to do so because of the inactivating domain that sticks right here and inhibit the channel function. And then only when you restore the cytosolic negative state back again, then this protein becomes again functional.
Can you explain the mechanism of the inactivation?
It's the inactivation mechanism that explains the refractory period. It didn't I mean, the atomic. You mean, what do you mean by explanation here? The atomic level description. Again, depolarization, which happens due to the action of.
What is this circle, white circle, and why it's blocking because of the positive charge?
Okay, so again, this is a schematic model, obviously, for the protein, for this voltage-gated sodium channel that's responsible for the rising phase. And now, so schematically, it has a domain filled with positive charges, and this is what we call the sensor domain. And about the globular domain, so why does it stick right here? Okay, so it's also because electrostatic interactions, which means it has a total charge of negative charge. And it has, again, some stabilizing interactions, and there are some crystallography that explains its stability right here in this location. There are some stabilizing interactions of this globular domain in the entrance of the channel.
Why the voltage sensor is positively charged?
I don't know why is it positively charged, but it's positively charged, and why is it pushed towards the exoplasmic side?
What is the function? What does it do?
What does it do? So, okay, it electrostatically being pushed away by the excess of positive charge during the rising phase of the action potential. So when the inside is negative, this one stays right here, and it holds the conformational state in a closed state. And when it is pushed towards the outside, that's the driving force for the conformational change in this protein that opens a channel.
And you said something about the albumin and
These positive charges are due to high concentration of these two amino acids in this region, lysine and arginine. You see the positive charge right here on the amino atoms. Okay. So that part explains the rising phase, and also maybe why the phase stops to rise at a certain point, which is again, as we saw, it's the binding of inactivation domain to the channel exit, which stop any further rising. But what stands in this second hyperpolarization or repolarization phase of the action potential, and that's obviously a brief schematic explanation or a reductionist explanation. But the main determinant for this phase is the voltage-gated potassium channels. So not only voltage-gated sodium channels exist, but also voltage-gated potassium channels, which upon further depolarization of the cell, of the membrane, would be more open or more active. Now, so this time it's a negative feedback. Sodium channels, voltage-gated channels, have a positive feedback intrinsic in them, right? Because when the membrane becomes more depolarized, they become more active, which causes more depolarization and more activation and so on. But for the potassium voltage-gated channels, more depolarization, which will cause more activation of the channel, would cause more hyperpolarization, because potassium ions, which are positively charged, flow outside of the cell, towards the outside. And so the membrane, the hyperpolarization state of the membrane is restored by the function of these proteins. And because they are open after the sodium channels, they are sometimes called delayed potassium channels, and their currents are sometimes called delayed potassium currents. So what do you think? If both these channels would have the same kinetics, they would be open and activated at the same rate, what would happen to the action potential? Sort of neutralize it even, right? So you have this delayed reaction of the potassium channel, both of them with the same dependency, the voltage dependency, as the sodium channels. They have to be delayed for the action potential to be formed. Okay. And these are some examples for patch clamp traces, where you clamp a tiny fraction of the cellular membrane on the tip of an electrode, which allows you to record a single ion channel or a small amount of ion channels, just several ion channels. And it's a voltage clamp. You hold the membrane in a certain voltage and record currents. So the units here are picoampere. And you hold and you record the currents of the potassium voltage-gated channel in different voltages, in -10 and in +50. So first, what are the changes between this state, the hyperpolarized state, and the depolarized state? In the action, okay. So again, what's the difference in the action in the function of the potassium voltage-gated channel when you hold it in this state, in hyperpolarization, or when you hold it in depolarization? What changes do you see in this trace compared to this trace? So first you see that the difference, the amplitude is different between the two states. Amplitude, just the height of this block here compared to this one. Why is the amplitude increased in depolarization? Why? Sorry? Okay, so more channels are open, but here you see the function of only a single channel, because you patch clamp the membrane.
So they move faster?
So they move faster, right. Why do they move faster? So this description, as I was requested, does not include any equations, but this is really not anything. So the current of a specific ion is the conductance of this ion multiplied by the driving force, koach electromagnetic. The driving force, that is the difference between the battery of the ion and the voltage, the current voltage of the membrane. So, and the battery for the potassium ion is down there in -80. We've mentioned it. So, as you see, if you hold the membrane farther away from its battery, from the specific ionic battery, then the driving force increases, increases together with it the current of the ion. So the first change is the amplitude. What about any additional differences you see between the hyperpolarized state and the depolarized state? Well, number of times of frequency of occurrences. So more opening events occur when you depolarize the membrane. You even see some cases in the depolarized state of two channels open at the same time.
Even though you said that's what confused me, because
Okay.
you said you record just one channel.
Yeah, okay.
It's just like two.
So just a couple of channels, because sometimes you patch clamp a fraction of the membrane that has more than one channel, but that's that.
Okay, so it's supposed to be one.
Yeah, and it so sometimes you get two open at the same time. And the last thing is the length of when a channel is already open. So how long does it stay open? In the depolarized state, it stays open longer than in the hyperpolarized state. And these three features, in general, push the channel towards a more active state in the depolarized state. Okay. There are plenty of known voltage-gated potassium channels in animals. Again, just the fact to appreciate how important this protein is. Okay, so the function of these two proteins, the voltage-gated sodium channel and the voltage-gated potassium channel, together can explain the propagation of action potentials along the long projections of neurons. So let's consider a fraction of time, a freezing time point along the propagation of action potentials in this, for instance, axon. So this is the side of the axon hillock where the action potential is generated. The axon hillock is where the soma becomes, where the soma becomes an axon. So this is the region in the neuron that is called the axon hillock. It has high concentration of voltage-gated channels. This will be the dendritic tree up there. So this side of the figure is the axon hillock, and the action potential is propagated unidirectionally towards the terminus. So why unidirectionally and why without diminution? And the reason is that, okay, so in a given moment in time, you have, in this region of the membrane, you have depolarization. Voltage-gated sodium channels are hence opened, cause a flow influx of sodium into the cytosol, which causes more depolarization, which causes more activation of the channel. So this is what happened in this fraction of the membrane. But right here in the back, where the action potential have just left this area, you have inactivation. You have voltage-gated inactivating channels, non-activated. Because for a given second, we might think that this propagation would happen to both sides, towards the axon hillock and towards the terminus. Because here you have voltage-gated channels, and also here you have voltage-gated channels. But these two population of voltage-gated channels are in two opposite activation states. These are in an inactive state, and these are in a standby active state that upon slight depolarization will open and cause the positive feedback. So an action potential, while it propagates towards the terminus, it leaves behind it, so to speak, a tail of inactivating voltage-dependent channels. And that's the reason for the unidirectional propagation of action potentials. And again, also, what exactly is this period? How is it connected to the structural model we showed previously? So this is, again, the refractory period. Right? This is the region in space or in time. You can talk in these two levels on this mechanism of the inactivating ion channels.
What is diminution?
Decreasing. Hamata. Haktana. Becomes smaller. Okay, so now, and another thing that, again, action potential are propagated without diminution, because in every given moment, they sort of being created or produced back again from anew, because again, the action potential is where is these, or the peak of the action potential is where the, sorry, again. It's a wave moving, it's a disruption moving in time, and the disruptions of the ionic gradients across the cell membrane. And each time this active process of the voltage-gated channels give a boost for the action potential, and that's what gives us the propagation without diminution until it reaches the terminus of this projection. Okay, so this is the distribution of voltage-gated channels that allow this kind of propagation. Nerve cells can conduct many action potentials in the absence of ATP. It's a classical experiment when you cut one leg of the squid, or one arm, I should say, of the squid, and it still can propagate action potential. And the reason is that, as you see, that you don't use ATP in this mechanism that I've just explained to propagate action potentials. And let's so let's consider some numbers that will even better explain why you don't have to have ATP for long durations, and still neuronal projections would be able to propagate action potentials. And we will get some intuition about the numbers of concentration of ions in the cell and so on. So typically we have around 10 voltage-gated sodium channels per square micrometer of plasma membrane. And if we will make a very reasonable assumption that each channel passes about 5,000 ions during the milliseconds it is opened, so we will get to a maximum of 10 to the power of 5 ions per square micrometer of plasma membrane during a single action potential.